Hi, I'm Paul Milne. I'm a postdoc researcher here in the Newcastle University. And quite a lot of what we do is using flow cytometry to try and find the cells which leukemia cells come from, where they originate. So flow cytometry uses a method of detecting the proteins on the surface of a cell based on fluorochromes. So you tag the proteins that you're interested in with a particular fluorochrome, run this through the cytometer, and it detects the emissions, so then you can differentiate the cells on just 2D scatter plots. We can then use this to sort out individual cell subtypes. The reason we want to do this is that quite a lot of cancers, such as Langerhans cell histiocytosis or acute myeloid leukemia, have specific mutations, and we can detect the mutations in cells using PCR. We can then look at which subtypes of cells have the mutations and try and work out their origins. Okay, so now each of these vials in front of me is specific for a different protein marker on the surface of cells, and they're all markers which could be relevant to the disease we're looking at. I'm looking at 4 different patient samples of an acute leukemia where after treatment for the leukemia subtypes change. So an analogy might be if you culled all of one type of shark in the ocean, another shark might then take its place. So this is quite rare for a leukemia to switch like this, but it's important we understand why this can happen and how, so that we can try and monitor patients for this happening and get to them early to give them treatment for their change in leukemia. So I'm just going to make up a master mix with all of the different antibodies added. Just going through in turn. We can add from the master mix to each of the samples. Make a master mix just to make sure that each sample gets the exact same combination of antibodies. Any differences that you can then say for certain is due to the disease rather than any technical issues. and then we'll just leave these samples to stain for 30 minutes. Okay, so this is an example of what we do here with flow cytometry. First we get a blood sample which contains white blood cells and red blood cells. From here we can use some lysis buffer to remove the red blood cells that our group aren't interested in. So then we have a pure population of white blood cells. We can mark these white blood cells with the different antibody markers that we saw before and pass them through a detection laser. The laser goes past and measures the expression of these markers, and you can see here on the 2D plots. You can then gate the different cells. So, for example, this green cell is positive for marker 2 but negative for marker 1. This orange cell is positive for both markers. You can gate off these cells. You can then use the cytometry equipment to alter the electrical charge on the cells based on their expression of these markers using the software. And then they'll be deflected either to a negatively charged plate or a positively charged plate and sorted into different vials. And what we can look for in this is a number of different conditions. We can look for increased numbers of a certain cell type that you would see in a malignancy like acute myeloid leukemia. You can look for changes in the cells which you'd see in functional disorders. You can look for lack of cell types, which we see in GATA2 deficiency, or you can look for very subtle immunodeficiencies where very tiny subsets of cells have changed in number. So this is the actual fusion cytometer which was funded by Bright Res, and this is what we're going to use to sort the different cell types from the patient samples. So if we just look at the screen, we can see examples of the data that we get from the samples. So each of these dots on these plots is a single cell going through the cytometer. We can then use the plots to narrow down the cell types that we're interested in. And then using the very specific surface markers, we can gate the cells and then tell the cytometer that these are the cells we want to sort. On the right hand side we can see a visualization of the cells actually going through the cytometer. Each of these again is one cell within a droplet of liquid passing past the detector to then where the cells will be collected. And back onto the machine, you can see that the sample would be inserted on the right hand side here. The cells would individually go through the cytometer, and then based on the electrical charge which has been set up by the software, they'll be split into individual collection tubes. You can then take these cells and process further down the line for mutations.